This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 79 for April 23rd, 2010. Hi, everybody. I'm Vincent Racaniello, and this is TWiV, the podcast where we talk about the week's news in virology. And joining me today to do that from Western MA is Alan Dove.
Pleasure to be back.
Where'd you go?
Well, I
Just
was recording TWiV last week, and you hadn't talked to me since then.
Have you been home the whole week? Have you traveled?
I haven't. I don't get out much.
No meetings to cover.
No.
You stay home a lot. It's kind of nice. I saw your article or your coverage of the New York Academy of Sciences on, what was it, circadian rhythms?
Yeah.
It was pretty cool.
Yeah, that was a fun conference
and
too.
For that one, you had to come down here into the city.
No, actually, I do for the Academy. I do frequently remote coverage where they'll post the video, I'll FTP it, and I'll watch the conference on video.
Real time or after the fact?
After the fact. That's
a good gig, I would say.
Yeah, it's pretty nice. Some of the stuff I have to go to. Which is, I guess it's good to get out of the house occasionally.
So tell me, how do you, maybe this is useful for people to know who are thinking of a writing career, how do you resist the temptation as the weather gets warm to go out and plant a garden instead of working?
Well, actually, you busted me. I was just out today planting some of the plants for the garden. But
I guess you can work at night.
Yeah, the thing is the schedule, you have to set yourself a schedule. Anything where you're working alone or working at home, you have to say, okay, from approximately 9 to 5, I'm on the job and I'm not going to be goofing around or doing work around the house. I'm going to be working on actual work. Mm-hmm. But I've been doing this long enough that I have the self-discipline that I can go out and, plant a couple of plants in the garden for 20 minutes and then come inside and get back to work.
You have to be disciplined, right?
Yes. Yeah, it is not a good thing for people who really need a lot of structure.
It's easy to get distracted in today's world. If you have high-speed internet, that's it, right?
Extremely, yeah.
Or outside, that distracts also, yeah. Well, that's great. Well, I have to come in here most of the time. I don't have the opportunity to stay home. Someday, I could sit home and do podcasts all day.
Yeah, that's true.
It would be fun, but I wouldn't make a living, so.
No. So far, podcasting doesn't pay the bills.
For most of them, but maybe someday. Or maybe maybe it goes along with some other activity like writing books. You do podcasts, you have a name made, and then you write a book and it sells. I don't know.
Right.
Anyway, today we have a couple of neat stories. Alan and I will discuss them. And just in case you're wondering, Dixon de Pommier is on his way to the Canary Islands for a vacation. I don't know why he'd want another vacation. He just went away in January, but
that guy is always going someplace cool.
Well, he's retired, so I guess he can do that. Are the Canaries cool? I don't know anything about them.
Oh yeah, they're supposed to be gorgeous.
All right. Well, we hope he has fun. We won't see him next week, and I think the week after, I'm not sure. He's a traveling guy. Yeah. And Rich Condit is recovering from a little surgery at home. So he'll be back next week, and so it's you and I, but it's no problem.
Yep, we can handle it.
First story I've been meaning to talk to for a few weeks. It's based on an article in Nature News, which is accessible to everyone. Yep. And it's good considering the topic of the article. Yeah. It's entitled, U.S. Seeks to Make Science Free for All, and this is not virology, but it's science and science publishing. And we've talked about open access a lot here on TWiV and the idea that most of research gets published in journals that are walled gardens. You have to subscribe to get to them. And there's been a growing movement to try and open this up. And one model is to have open access journals like Public Library of Science. And they try and recover their costs in other ways. I guess they charge fees.
Right.
And they have some advertising.
Right.
And apparently that's working for them, right? They're going to be in the red at some point or in the black? I mean,
Well, they keep saying they're going to be in the black at some point. The PLOS is a really neat idea. I know most of the staff, I think, and I've worked with them at other publications, and then they went off to start this new public library of science thing, and it's gotten off to a great start. But when you look at the cash flow in it, um, I don't think they've yet proven that this is a sustainable business model.
I see.
Um, and there was Nature actually, you could say that this was motivated by a particular agenda, but they did an investigative report on where the money comes from and goes to it in PLOS Publishing, and they determined that in fact their lesser journals, PLOS One in particular, which does a very quick peer review and publishes things and then allows subsequent comment on the papers. That is apparently the major cash cow,
Hmm.
because the way PLOS works is the author pays to publish their work in it.
Yeah.
So the more papers you bring in and the more papers you publish, the more money you make when you're publishing that way.
Hmm.
This, of course, produces a motivation for the publisher to publish papers that might not be so great. I'm not smearing the papers that have come out in PLOS. Actually, I think there's one that we're going to talk about today. There's excellent work coming out through that. But if you want to expand this model and say, oh, the whole science publishing industry could go this way, I think there would be certainly operations that would just publish anything at all in order to get the author's fees.
Yeah, that's a good point. When you link it to survival that way, it's a problem, right?
Yeah.
I mean,
Yeah, I mean, this has been as a scientist turned journalist, I'm torn about the whole open access thing. I really, really want to see it work. Because as a scientist, I think that the more access people have to science, the better. But as somebody who pays his bills, by checks that are written by science publishers, I have to ask, where does the money come from?
no, it's absolutely correct. I mean,
And
PLOS doesn't
yeah,
publish, they publish a fraction of the entire literature.
right.
I mean, there are a few other open access journals, but I don't think they're as large as PLOS. So most of them are still published behind walls, and they will be until someone figures out how to do it sustainably.
Right.
And I think that's a good point that it's not been proven yet whether it's going to work. We'd all like to see some kind of open access work, but we're not sure because it costs money to do all of this. Right?
Yeah. Yeah, and everybody points out, oh, well, you don't have to print the journals anymore. You can distribute everything online, and it's a lovely idea, but it still costs money. And when you have review articles and news articles that are value added that people like me write, that costs money. And archiving this stuff, something that often gets lost in the shuffle is we're not just talking about news about the latest celebrities here. We're talking about the scientific heritage of mankind.
Right.
No,
No,
that sounds kind of high flown, but that's actually what we're dealing with in archiving this information. And so when you say it's not going to be printed, it's going to be stored digitally, is that really wise?
Mm-hmm.
And I would love to see, as I say, I would love to see this work. I just am worried that 30 years from now, somebody's going to try to look up a paper that was published way back in 2010, and it's going to be, on the future equivalent of a 5.25-inch floppy disk. It just won't be accessible.
Well, it's sort of already happened. I mean, our library here has gotten rid of a lot of old bound journals.
Right.
And many of the old journals you can't find online.
Right.
They're just not available. You go to PubMed and you don't even get an abstract.
Right.
If it's before the 60s, more or less.
Yeah.
We used to be able to go downstairs and get them. So it's its own obsolescence right there, right?
Right. Right. But at least in that case, the paper version of those papers exists somewhere.
Yeah.
So yeah, it would be additional work, and you'd have to bring it in from the depository or wherever they've crammed those away. But sometime within the next couple of weeks, you could probably get your hands on a copy of that paper. But that is an issue.
Yeah. Well, that's one approach, open access, and the other one is what this article is about, that the government is trying to make regulations such that if you receive government funding for your research, then you have to make it accessible at some point. And this was originally set forth in 2007, that Congress passed a bill saying you had to deposit your paper in PubMed Central within 12 months of publication. It was originally voluntary, but that didn't work, and then they made it mandatory.
Right.
And now there are quite a few papers there. I just checked, and actually mine are there. Someone must send them for me because I didn't put them there myself. But they're all there, and you can get there's a direct link to a download right at PubMed Central.
That's right.
2 million articles, 420,000 downloads a day.
Yeah, and I use that as a tool when I'm researching stories. I mean, I have access to a number of journals, but not all of them.
Yeah.
So if I'm researching the background on something and I find a paper, if it's in PubMed, and it's in PubMed Central, that's wonderful because I can just grab the full text and read the information.
Right. So now there are two bills on the table, I believe, which is basically going to cover all federally funded research.
Right.
And I think they're going to shorten the time to 6 months as well. And you have to deposit some version of the paper within 6 months.
Right.
And I think it doesn't have to be the final version that's published and all nicely set up. It could be your manuscript, a PDF that you deposit, I believe.
Yeah, I think they're still working out the details on that, which, of course, raises another issue, and that is who gets to see the peer-reviewed version and who gets to see the...
Yeah, it can get complicated, right?
Yeah.
Now, as you might expect, there's a quote here from someone who's a director at Elsevier.
Yes,
who's a huge journal publishing operation, and he doesn't like this. He said it's overly government regulatory. Shouldn't be messing in the business of scholarly publishing. But of course, he's biased because he works for the company, right?
Well, and I should disclose, I've done a substantial amount of work for various Reed Elsevier subsidiaries over the years. They're a huge publishing organization. He's got a good point. This is a government intrusion into a private business. But at the same time, it's government-funded science that's on the table, so the taxpayers do have some claim on being able to access the data that they effectively paid for.
Right. Now, there's another issue here, which you've probably heard about, this Compact for Open Access Publishing Equity.
Right.
Which has been. Do you know any more than I do about that?
Not really.
So it's basically starting in September 2009. A number of institutions have signed up, including Columbia. I think Harvard is another one. And they contribute some funds, and they assure that their publications are going to be provided ahead of publication.
Right.
And that's probably in a pre-publication format, just a manuscript. Now, here at Columbia, when you submit a paper, there is a website you can put it on. It's not open to the public, but everyone within Columbia can see it when you submit it. And then when it's accepted, you can open it up to the public. That's part of the Compact idea.
Right.
And actually, I have a contact at PLOS who has agreed to come on TWiV and talk with us about this. She's involved in this Compact. So they're working beyond what they're doing. They're working beyond the open access model. They're trying to work with universities to get these individual models to work, I suppose, which is good. I also found this week an article at taxpayeraccess.org, which is the federal government's bill to do public access. It's called Federal Research Public Access Act.
Right.
And it's basically the act proposes to make manuscripts reporting on federally funded research publicly available within 6 months of publication in a journal. So this is up for review in Congress at the moment. And if you go to this website, we'll put a link in the show notes. You can, contact your Congress people and tell them what you think about that and so forth. So it's got a lot of resources about this whole thing. I think Lieberman was one of the proponents of this bill.
Yes.
as well as another representative or someone from Texas whose name is evading me at
Yeah,
this
it
moment.
has bipartisan support.
I think it's a great idea. I think it's a good
Yeah.
start, and because I do think that a lot of people can't read the basic research. It's not accessible to them, and they would benefit enormously. And maybe we get great questions here, but maybe a lot of them would be answered if people could access the papers.
Maybe, although I think the going back to the value added for a lot of these journals, people would benefit more from reading sort of the top level reviews,
Hmm.
which are not on the table for public
No,
access.
no, that's going to be behind the wall. You're right. Yeah, often those are the great summaries. Yeah.
Right. And maybe that'll eventually become the business model, that you can get public access to the basic research papers, which are going to be hard to read because it's very inside, very technical stuff. But you might pay for access to the more broad summary type stuff that would be more interesting to a lot
Hmm.
of folks.
Oh, maybe the journals should consider fee models for the general public, non-scientists or non-professionals, non-researchers, for example.
Right.
A retired person wants to just have a subscription to Nature Science Cell.
Hmm.
Give them some kind of a break. You don't have to show them everything, maybe the reviews and so forth
Right.
at a reduced rate. Well, anyway, that's their problem to figure out, but we do the research and we'd like everyone to see it, so I hope this goes forward.
And the model that I would love to see work out eventually, part of the problem is the traditional journals are very heavily invested in the print medium still, so they have a lot of the same overhead problems that the newspaper industry has. Um, but when we do finally get to a point where they can move out of that, it would be great if you could have advertising supporting the websites, um, which would address a lot of these issues. If you can get enough advertising revenue to support these things and make them free, I think people would benefit tremendously.
Yeah, I agree. All right, let's move on to the second story, which you picked up a couple of weeks ago, having to do with the eradication of poliomyelitis.
Yes,
I think we discussed this on and off over the months on TWiV,
I
right?
seem to recall that may have come up at some point.
Why don't you go ahead and tell us what you found?
Yeah, so the story that initially caught my attention was, there's a kind of a think piece in the Lancet, April 15th edition, um, and it's a couple of ethicists who've written an article saying that the world has an ethical obligation to finish the job of polio eradication.
Mm,
Um,
right. Yes.
and this is an interesting claim. Um, and they do make some very good points. It's a very persuasive case from an ethical standpoint. The first thing they do is set aside the economic arguments, which I'll do for just a moment, but then I think we need to get back to them. And they point out a lot of this discussion has centered around the costs of eradication and what that trades off against. But their approach is to just look at what is the impact of not eradicating the disease versus eradicating it, and ethically considering the possibility that we have the power to eradicate it, which may be a safe assumption. I'm not sure. Um, but they then conclude that the human cost of failing to eradicate polio is um, millions of lives destroyed through the generations, even if we're continuing to try to control the disease. If we haven't fully wiped it out, you've got all of these additional cases piling up, and they conclude that this is ethically inappropriate.
Hmm.
And it's an interesting article. They do, as I say, they make some very persuasive points if you look at the piece solely in its own terms as an ethical argument.
Right.
The problem I have with it is that I don't know, ethically, I think I'm a pragmatist. I look at the world as it is and try to figure out what solution makes sense. And unfortunately, the world as it is operates on an economic basis, fortunately or unfortunately. And in public health, if you're spending billions of dollars, and I think one of the articles you pulled up on this actually has some very interesting figures on it. Um, we've spent over the course of this drive, they've spent about $8.2 billion, and that's money that a lot of it has come from other public health programs.
Yeah.
Because unfortunately, that's the way public health programs work. It's not like defense spending where you can just write an unlimited check. If you're spending money on one disease, the money is coming from control efforts on other diseases. It's a zero-sum game. Um, so we're stuck living in this world, and I think while the ethical argument is interesting, ultimately I would still come down on the side that this is not the best expenditure of public health spending.
It's worth pointing out that this is probably all stimulated by the fact that April 12th was the 55th anniversary of the first polio vaccine.
That's right.
The Salk vaccine, yeah. So there were a couple of articles on this. And then your other article said trying to eradicate a disease is a waste of money.
Yes.
It's an article published in the Proceedings of the Royal Society.
Right.
Which is what you have just said, that the money can be better spent on other health problems.
Right.
Because even though we're very close to polio eradication, and close is a difficult word. I mean, we're much closer than we were in 1988 when the eradication program started, but the last bit may be incredibly hard. It's not clear yet that we can do it. There's so many issues, and even though there are only 5 or so countries with endemic polio, that is circulating wild-type polio, it's been very difficult to get it out of those countries.
It's like most projects. The last 10% takes 90% of the resources.
Yeah, the problem is that...
well, there's the allocation problem that I was just talking about, that this draws money from other programs. But there's also the issue that this becomes a harder and harder job the smaller and smaller the population. Ironically, the smaller the population you're trying to reach, the harder the job gets.
Right. Here's what I was thinking. So right now we're doing massive global immunizations. And if you say, okay, we're not going to eradicate, we'll just leave it the way it is, you're going to get massive outbreaks again because these massive programs will not continue. Right. You'll go back to standard healthcare, which probably doesn't mean a lot of immunization in a lot of countries. And you'll have outbreaks of the disease more so. So you're kind of stuck. If you've gotten people used to fewer cases every year, and if you went back, the whole thing would be perceived as a huge failure. It would probably influence any future attempt to do a similar thing.
Right. But I think, the argument in this second article is actually much more general than just polio, which is it's an economic analysis showing that attempting to eradicate any disease is probably not a good bet.
Yeah. That's from a cost perspective.
From a cost perspective, which, it's a cold calculation, but I think that's the kind of calculation we need to make here. And really, when you talk about going in and eradicating polio in an area, a lot of what's going on is these teams will go in and they focus on vaccinating everybody against polio. And then, the population that lives there is still dying of malaria and tuberculosis and AIDS. And you haven't dealt with any of these other health problems because you've been so focused on combating polio. And to their credit, the WHO has made efforts to try to use this as a way to build more public health infrastructure worldwide. But I think making one disease the focus of the whole thing is just doomed.
But I don't think they're going to change their course, do you?
No, there are too many careers on the line at this point.
Well, a lot of money as well, and Bill Gates is really committed to this.
And it's like, have I told this joke about the fellow driving through the countryside, and he really, really has to use the bathroom, and he sees a farmhouse with an outhouse outside it. So he figures, oh, perfect, I'll just run up there and use the outhouse. He pulls over, he runs up to the outhouse, he throws open the door, and the farmer is in the outhouse and is completely shocked, and jumps up and all the change flies out of the farmer's overalls pocket and falls down the hole in the outhouse. And the farmer has got his pants still around his ankles, and he looks at the traveler who's thrown open the door and shakes his head. And the farmer reaches into his other pocket and he pulls out a $20 bill and throws it down the hole, down the toilet. And this fellow looks at the farmer. He says, "Wait, wait, wait! I'm really sorry that I startled you and made your change go down the hole, but I don't understand why did you throw that $20 bill in there?" And the farmer looked at him and said, "Well, you don't think I'm going down there just for change, do you?"
Excellent.
So,
Very good. Well, Bill Gates is heavily invested, as you know.
yes, and he's not going down that hole just for change.
He's not. He's
So,
he's put a lot of money into polio. In fact, I have an article from the Wall Street Journal, which seems to be accessible to
yeah,
everyone. I got the whole article here. I'll put it in the show notes, where it's called Gates Rethinks His War on Polio. He went to Nigeria last year, where there has been an ongoing outbreak because they stopped immunizing a number of years ago. They've resumed in the meantime, but it's very hard to recover, and it's spread to other countries. At the current moment, there is an outbreak in Africa in a number of countries where it had been eradicated. It all probably spread from Nigeria. So he went down there and talked to the Sultan, and they said, "Yeah, we're gonna lick this." So I don't know what is his change, but he just was told that it's not as easy as we thought it was going to be, and we
right.
need more money, basically, Bill.
Yes.
So I think he will put more money into it. He's got $34 billion in his charity.
Right.
It says here in this article, a big hurdle is money. The polio program is $1.4 billion short of the $2.6 billion it needs over the next 3 years. The Gates Foundation will continue its polio grants, but says it can't make up the shortfall. So it's all about money.
Yeah.
Anyway, so that's an interesting article. Then there was also an article in the Times this week, which basically is saying it looks really good. I don't know why they're doing this. The number of cases this year is much lower than it was at the same time last year, particularly in India and
Right.
in Nigeria. And this is stimulating optimism left and right, which isn't really the right thing to do.
Right.
Because who knows, these things can change. So just to give you an idea of the numbers, there's a great website, polioeradication.org, which gives you all the current numbers. And what this article in the Times fails to tell you is that there's an outbreak in Tajikistan, the first importation since Europe was certified polio-free. So this country is now has a little outbreak of polio, and it had been declared not to have polio anymore.
Right.
Okay, so that's one thing. Then if you look at the case counts, let's go. You can go up to global situation, global case count. This year, to date, 2010, globally, 71 cases of polio. And this same time last year, there were 328. So it's gone down so far quite a bit. I don't think it's going to remain at 71.
Right.
It's quite a bit down in India compared to the same time last year. Nigeria is 2 so far this year and 193 last year at the same time. India is about half, 19 versus 32. So there's all this optimism. I don't really know why. Do you understand that?
Well, it depends on whose numbers you're looking at and how you're analyzing them. You know, the good news is that the cases are down in a lot of places that had been hard to deal with in the past. The bad news, of course, is that the disease is still around, and this outbreak in Tajikistan is certainly not good. And I think we should point out, we talk a lot about how we've got this drumbeat of doom going here for the polio eradication campaign. I would love to see this work.
Yes, I agree with you. Sure.
Please, I hope I'm wrong, but I haven't been so far, and the odds don't look good, and I don't know that we should keep throwing money down this hole. So what the Times is trying to do is they're reporting the trend. They're reporting that this year in some of the areas that were the hardest to deal with in the past, the numbers suddenly look great. And I say that's wonderful, but let's also, you know, keep the real perspective that this is not gone, and we still have the issue of the vaccine-associated virus because of, we've got all these people who are going to be chronic secretors that we now know about. So how long do we have to keep vaccinating to keep this disease gone?
No, that's the big deal. I mean, we're using an infectious virus, and we know it reverts in the alimentary tract. So even if there's no more wild disease and everyone's immunized, when do you stop? Because when you stop, you now have new susceptibles who will get infected with this vaccine-derived virus.
That's right.
This is a huge problem. And in connection with that, I found a paper from India and WHO where they basically show that the gut immunity conferred by the vaccine isn't as good as we thought it was. They did a study in India where they looked at contacts of people who had polio. In India, you can find plenty of those.
Right.
And then you look at their contacts who have been immunized. And what they find is that about 20%, up to 20% of the contacts are shedding wild-type polio from their intestines, even though they're immunized and protected against disease.
Right.
And they say, although OPV is protective against infection with polio, the majority of healthy contacts who excreted wild-type polio were well vaccinated.
Were they vaccinated monovalent or trivalent?
It's a good question. Let's see. 12 or more monovalent type 1, yes.
Okay.
Some of these kids had 12 doses of monovalent type 1, which is the other problem in India, that 3 doses, which is what you get here in the U.S. and many other countries, is not enough.
Right.
You need 10, 12 or more. What a logistical nightmare.
Yeah.
12 doses. Anyway, yes, this is monovalent type 1 OPV. And they still will shed polio if someone walks by and infects them with wild-type polio.
Right. So all these vaccinated people they won't get the disease, but they can be carriers.
Right. So this means the herd effect isn't so great as we thought it would be.
Right.
So you're not protected in a community or a region just because most of the region is immunized. They're still spreading virus. And we don't know if this is going to be a problem, but they say at the end of this paper, we need more studies to figure out if this is an issue. So here we are, towards the end of an eradication campaign, and there are fundamental issues we haven't sorted out yet. And they say, maybe intestinal immunity wanes very quickly after immunization. That could be a tragedy, right? So
yeah.
It's a very interesting article and very troubling. I agree with you, though. I wish it would work. I just have this feeling it's not going to be very easy. And easy meaning years and years and years. It's going to take many, many more billions of dollars, I think.
Well, and the initial projections were that this would be done for the year 2000. And those were delayed projections. And, so here we are. It's 2010, and we're still talking about this.
I think that our original idea to use the killed vaccine is a good one, but I admit that we don't know if it would work in Uttar Pradesh in India, where you need 10 to 12 immunizations. And it's an injection. It's problematic.
Yeah, but the issue is, if you can't deliver an injected vaccine, a well-established injected vaccine, then you don't have a basic public health structure in place.
That's right.
And that's the underlying problem that we should be dealing with is the general issue that most of the world's population does not have access to even rudimentary medical care. Unfortunately, that is a decidedly unsexy issue, whereas eradicating a disease is a very sexy issue. So,
but could you do that for $8 billion globally in the countries that need it? So that's
you could not, but you could make a dent in it.
Yeah.
There was I was at a meeting a number of years ago, like 5 years ago, I guess. And there was a presentation by I've now blanked on his name, but he was directing the Global Fund for AIDS, TB, and malaria. And he projected that you could make a huge dent in controlling those 3 diseases, which those are huge, for about $10 billion. And he admitted that's an enormous expenditure in public health terms, but if you frame this as one of the biggest problems facing the world, which it is, then that's not such a huge expenditure. This is within the means of governments in developed countries to do, and it's in our interest, just for the security benefits that this would give us. But that's not where the emphasis is going.
Yeah, well, that's the key here. That's the discussion that was brought up by that first paper.
Right.
And eradicating sounds great, but what's the price in the end that you're paying for it?
Right.
And we have one other story to do, but before we get to that, I'd like to acknowledge the support of TWiV by Data Robotics, the makers of Drobo, which is a storage solution for your computer. One of my favorites. I love the Drobo. And now they're making, when I first bought a Drobo years ago, they had one model, the Drobo, which had 4 drive bays. These are enclosures you slide hard drives into, and they're very flexible. You can put any size, any speed hard drive. You plug it into your computer and put your data on it. And it's automatically backed up along the drive array, so that if any drive fails, you just take it out and replace it. And now Drobo has expanded its line. They have a Drobo S, which is a 5-bay model. They have a Drobo Pro, which has 8 bays if you are doing a lot of data-intensive work. And there's a Drobo Elite, and now a Drobo FS, which connects to your Ether network and make the data accessible to everyone. They're really fantastic servers. So just this week, I ran out of space on one of my Drobos, and I bought a 1.5 TB hard drive, which you can get less for $100 now. Pulled out the old drive, slipped in the new one. All the data were restored, and now I have an extra terabyte hard drive, which I can use for something else. So I love these things. I have them at work, and I have them at home. Now, Drobo, of course, you can, if you're interested in buying a Drobo, you can get $50 to $500 off, $75 to $500 off, depending on the configuration, by going to drobo.com/TWiV. And there you will see the instructions. If you use the coupon code TWIVPOD, all capital letters, you'll get an instant rebate from $75 to $500. Depends on the model and whether you get any drives with it. But if you have data that's worth the while, and most of us do, check this out. It's a wonderful solution, and I wholeheartedly endorse it. I love this photograph on the page of all these drives stacked up.
Yes.
It's great. Drobo, you'll love it. And we thank Drobo for their support. We're giving away a third one, and the contest rules are being developed by Data Robotics, so we're waiting to hear from them. Our last story for today, it's based on a paper in PLOS One. It's called Pepper Mild Mottle Virus, a plant virus associated with specific immune responses, fever, abdominal pains, and pruritus in humans. It's a hard one to pronounce.
Pruritus. Oh.
This is a plant virus. Pepper Mild Mottle. What a great name.
Plant viruses are so great for that.
It's a plant virus, and it infects pepper plants of various kinds. And apparently, very recently, it was found that this is a major part of the virus that you get when you sequence the virome in people's alimentary tracts. The pepper virus, which we'll abbreviate it as, was detected in stool of 12 out of 18 individuals in San Diego or Singapore. So what they did in this paper is to buy in France, so the authors live in Marseille, they bought 28 commercial food products at various grocery stores, and they tested them for the presence of this virus. And they all are products that would have, I guess, chilies in them or chili peppers. They have some that are controls, I guess, mayonnaise, oil, exotic sauces, spicy powders, green chili pepper, red chili pepper, yellow chili pepper, cayenne pepper, chili and cumin, curry, all these things. All right. And they tested them for the presence of the virus by PCR. And they did a little sequencing and a little electron microscopy. And they found the virus in 12 out of these. Let's see, what's the number here? 12 products were positive for the RNA.
21 products total.
Yeah,
12
12 were positive.
positive.
So over half. So it's a lot. And all the products were ones that had some kind of chili, which would be derived from a plant. So you figure this virus is on the plant and it gets into your seasoning or whatever. Okay, that's fine. But then they said, can this make people sick? So then they did a study where they got stool samples from adults and children, and they looked in that to see if there was the virus present. All right, so in children, they found RNA in one child out of 137. Not very much. But I guess kids don't eat much spicy food, right?
Right.
So that could be understood. Then the adults, the stool from 22 out of 304 adults were positive by PCR, 7%. Okay, and they could see particles in some of them by electron microscopy. And this virus is very much like tobacco mosaic virus. It's a long rod-like virus. So you can see in these EMs, it looks just like that. Then they said, well, these people who have this virus in their stool, do they have any symptoms? And then so they look at the records, and it turns out that there's a good correlation between having this virus in your stool and having abdominal pains, diverticulosis, diverticulitis, fever, and even pruritus, which is a rash of some sort. So it looks like I would have said, you eat chili, you're going to get a chili virus. But these guys say, we believe this provides the first evidence that plant viruses may cause disease in humans. In my view, of course, I think that it could be, but what's more likely is that you eat chili, you feel sick, or you get a rash because you're allergic or something, right?
And very best line in the results section, I think. Tabasco sauce contained the highest viral load, estimated to be nearly 10 to the 7th RNA copies per mil.
Right.
So these are spicy foods, and Tabasco sauce has the highest viral load, and I like it, but I guess if I had too much of it, I might get pruritus too.
You eat a lot of spicy stuff, you feel bad. It does things to your intestinal tract.
Right.
And the more you eat, the more virus they isolate, so the virus correlates with symptoms, right?
Right.
Doesn't mean that the virus is causing this.
Also, we don't really know what the denominator is, because this was stool samples from patients who'd shown up for other things.
Yeah, that's right. So there are a lot of variables here that need to be addressed. Now, they did find antibodies to these viruses in some of these people.
Right.
But if you eat enough antigen, you'll make antibodies. It doesn't mean that it's replicating in you in any way. So, I mean, it's an interesting idea. And in support of this, what they say in the discussion has been shown recently that tomato spotted wilt virus could replicate in 2 human cell lines, HeLa and diploid fibroblasts. So I said, that sounds interesting. So I went and looked at the paper, and this tomato spotted wilt virus is of a different family. It looks very different from the pepper virus in this paper. It's actually a Bunya virus,
right,
which also contains viruses that infect us. And it turns out that virus, tomato spotted wilt virus, first of all, if you take the virus and just throw it onto HeLa cells, nothing happens. You have to mix it with a facilitator that allows it to get into cells, because probably there's no receptor on the cell for the virus. And the only way this virus will replicate in human cells is if you first express the viral RNA polymerase in the same cells. When you do that, and then you add that tomato virus with a facilitator, that is a chemical that lets it get into the cell, then you see RNA replication. So that tells you that the machinery for duplicating the viral RNA is present if you provide the enzyme to do it, but you don't make particles. The virus can't get in. So I think it's a leap from that to say that this pepper virus could infect people.
Right. It does tell you that you should not transfect your own intestinal cells with the viral polymerase from plant viruses.
That's right. Or any
That's
virus.
Probably a bad idea.
That's right. So that's not going to happen, obviously. But
no.
Like anything else, who knows in the end what can happen? So I think probably this is not an example of a plant virus infecting people. But, in virology, weird things can happen.
Sure.
They cite an example that I just discussed in my virology class this week. The hypothesis is out there that circoviruses, which we've talked about recently because they are present in a rotavirus vaccine,
Right.
they may have come from a plant virus with the similar genome called nanovirus. The rep protein of circoviruses and nanoviruses looks like it's made from both viruses. So they share features. So the hypothesis has been submitted some time ago that this virus jumped from a plant into a vertebrate. Vertebrates eat plants, and maybe
Sure.
the virus could be selected to grow in them. So it's possible. I wouldn't rule it out, but the data here I don't think support that.
Right.
So I don't think that we should worry about plant zoonoses yet, plant virus zoonoses.
That wouldn't be a zoono. It would be a
What would it be?
botosis, botoses.
That's right. Zoo is the animal, right?
Yeah.
Yeah. What's the equivalent of zoonosis for plant viruses?
I guess it would be a botosis,
Botosis.
but nobody's ever had to use the term. And so
Alan, there has to be a title, an episode title in that.
botosis.
Figure something out.
Yeah.
Anyway, pepper mild model virus. There you go. Take a look at the paper. It's open access. You can read it for yourself, and maybe you could do an experiment. Eat a lot of pepper and see if you feel bad, and then let's do a couple of emails here. The last few emails from the virology class at Indiana University of Pennsylvania. And one is from Ben, who says, I really enjoyed your podcast about reverse transcription. I was wondering, what makes retroviruses like HIV impossible for our bodies to fight off? Are there any retroviruses that can be eliminated from the body?
Well, in some individuals, HIV.
Yeah. Yeah, it can be. There are many people, there are some people who don't get infected. I should point out that actually it's not a very infectious virus. It takes about 1 in 200 coital acts to transfer infection from a positive to a negative person.
Right.
It's actually been studied. So you can fight it off. It's just in some individuals you don't, and we don't understand why. Some people are non-progressors. They don't get sick.
Right.
And then, of course, there are many viruses that infect us, but never infect and never succeed. Not just HIV, but there are all sorts of other kinds of viruses that don't have the right receptors or the right proteins to work in our cells. And we don't know specifically what they are, but someday we might learn about it.
Well, the tomato virus polymerase would be one.
Yeah. So I wouldn't say it's impossible for our bodies to fight off. But the thing is, Ben, once the virus establishes itself, it grows in your immune cells. And those are the cells that you need to get rid of infections. So that's a problem. And then, as soon as your body makes antibodies to neutralize the virus, it spawns variants that escape that neutralization. And this goes on and on and on every few months. You make antibodies, and the virus changes so it's not neutralized. You make antibodies for that one, then another one comes along. So that's a big problem. And probably the same goes for cellular immunity as well. Quite a slippery virus. Amazing. Good strategy to infect your immune cells.
Yep.
Paula writes, I was browsing the chronic wasting disease website when I read that prions not only occur in the brain and spinal cord, according to TWiV 67, but also in the saliva, blood, muscle, urine, and antler velvet of symptomatic animals with late-stage disease. So being that the prion has been found in all mammals, including humans, we do have the potential to become infected by prion exposure if we handle or consume the meat of an infected deer. I think it's pretty unlikely, but yeah, the potential is there. It could. I took a little line from an article. The likelihood of successful interspecies transmission of prion disease is influenced by the degree of homology of the infecting prion with that of the host endogenous prion protein, and that's the species barrier. They did an experiment here where they take a prion from one species and put it in vitro with another protein and see if it can convert it. So they did a cell-free experiment with chronic wasting disease prion and human prion. They can convert the human prion into a pathogenic one, but the efficiency is 14-fold lower or weaker than the human or the cervid prion. And they say, well, we don't know what this means to be 14-fold lower in terms of human transmission, but it's not as good. So My feeling is don't eat the meat of a deer who looks weird, is sick, and has a wasting disease, right?
Right.
The likelihood is pretty low, but you're right. It could be in other tissues as well. So saying, be aware of the spinal cord and all of that might not help you in the end. Sarah writes, what is an enterovirus? I used to work on enteroviruses.
Yeah, I've heard something about those.
Enteroviruses are viruses. They are members of the picornavirus family, and Alan Dove did his thesis on an enterovirus.
Yes, I did.
Poliovirus is an enterovirus. And they are originally called enteroviruses within the family because they infected you via the alimentary tract. They're resistant to low pH. So poliovirus, echoviruses, Coxsackie viruses. But now, by sequence comparison, they've thrown rhinovirus into the enterovirus family, Sarah, to confuse things. And they're still calling them enteroviruses, but now we have rhinoviruses, which are respiratory pathogens. So I can no longer say they're viruses that infect your enteric tract because they're not. But originally they were small RNA viruses with icosahedral capsids, members of the picornavirus family. Heather writes, I'm responding to your question regarding the usefulness of podcasts as a learning tool. The traditional classroom provides a forum to discuss and explore new concepts, which gives depth to the learning experience. But I feel the podcast has several advantages as well, which makes them worthwhile. Students can get exposure from a larger pool of experts who bring their unique background and expertise. Material can be instantly repeated if a section is missed or not understood. The podcast can be interrupted if the student wants to check out links related to the topic, look up terms, or whatever may increase their understanding and interest. They can then return to the podcast at their convenience. Finally, there is the possibility of the two-way exchange of ideas, such as this email. Although this feature is not inherent in a podcast per se, it can provide the tools to open the door. And so that's great. It's a nice answer.
Yeah.
Michelle writes, I wanted to say thank you for answering my question about viral resistance to antiviral drugs. I have studied microbiology, but virology is a new field for me. I have learned about bacterial resistance and the development of new drugs that combat resistance. So I was interested in knowing about this for viruses too. As a side note, I learned from my veterinarian that the same thing is happening with certain parasites and drugs used to kill them. To answer your question, I think that podcasts are an effective way to learn. I have only listened to TWiV, but I enjoy listening. The one thing I would caution is that if podcasts are used specifically for student education, that the presenters would have to be careful to stay on topic because it may be easier to stray when not in a classroom setting. Yeah, I thought that was interesting. We stray sometimes.
Yes, we do, but we're not strictly a class teaching exercise.
She's right, though. I try not to stray in the classroom.
Right.
Because you have a limited amount of time and you want to stay focused. Ken writes, I'm reluctant to be seen as chastising such a distinguished group of scholars, but I know
Go right ahead.
That's fine, no problem. But I know that you're committed to basic virology education. Toward the end of the episode, you all handled a question about what distinguishes the most complex viruses from the simplest bacteria. Listening to the discussion, I was dismayed that I did not hear what I regard to be the critical word. That word is eclipse. Well, you're absolutely right. I don't know if we were looking for that kind of difference. And I don't remember the question now, but you are right. Eclipse is a big difference. When you put a bacterium in culture medium, it immediately starts to divide. And you put a virus in a cell, there's an eclipse period where the infectivity is gone. And that's because the genome comes out of the capsid. You remember this, right, Alan?
Sure. Yeah, I paid a little bit of attention to that.
And this confused the earliest virologists. They didn't know what to make of this because they were used to bacteria and not having this lag period. But then Delbruck and colleagues started doing one-step growth curves and figured this out. That's the eclipse. So you are absolutely right. It is a fundamental beginning aspect of virology that we teach everyone. So thanks for telling us. David writes, I believe in TWiV 70, where someone asked the question, is there a virus that alters human behavior to facilitate transmission? Would rabies qualify? Yes, we've gotten two other suggestions for that, for rabies. Yep. And then he writes, I recall years ago speculation that rabies was the basis for the vampire legend. If I remember correctly, rabid people would try to bite other people, spreading the infection. If that is true, and if rabies can be spread by biting, then rabies could be considered a virus that spreads through altering its host behavior. I don't think rabid people are in any position to bite anyone.
Not in the late stages of the disease, and they do tend to, aggression, I think, is one of the common hallmarks. But
I think among animals, though, a raccoon or a bat, certainly their behavior has changed, and we had some good emails about that previously. But I would think not with people. I think late in the disease you're debilitated, but early on, I've not read any case reports of that, but I could be wrong. So if anyone knows better.
I don't think there are any case reports of person-to-person rabies transmission. I'm not finding any right now. It is certainly not common. I think we can safely conclude that. But rabies does change the behavior of a lot of animals. Certainly changes the behavior of humans who are infected, but it changes the behavior of animals that are infected in ways that would make them more likely to come in contact with additional potential hosts.
Yeah, so we don't find any examples of human-to-human transmission, but certainly the animal behavior has changed. So if anyone knows differently, let us know. Well, let's do this last one from Vincent, which is not me, by the way. I'm not writing a letter to TWiV. It's another Vincent. Thanks for the wonderful podcast. TWiV and TWIP are at the top of my podcast favorites. I recommend them to everybody and have posted a thread at Student Doctor Network, a community of pre-med medical and professional doctors. Great. A few comments and questions. One, you have mentioned several times that fugu fish have a compact genome virtually free of junk DNA. First off, does this include line or retrotransposons or just endogenous retroviral material? Well, actually, I looked this up, and I'll post a nice review article about this. But basically, they do have repeated elements. So I don't know who said that. Was that Dixon?
I might have said that. They certainly are. They have a lot less junk.
Yeah, they have a lot less. That's basically it. So I have a nice table here from the original paper, the science paper where the genome was sequenced, and they have a lot of the same repeat elements. They have line elements and other transposable jumping gene type elements, and they're just reduced in number compared with mammalian genomes or others. So, yeah, they do have pretty much what we have, just in sometimes orders of magnitude fewer copies than mammals.
Yeah, their genome is 1/8 the size of the human genome, but seems to have about the same number of genes, and it's the shortest known genome of any vertebrate species.
Right. So he says, I don't understand this. How could they have none? So they do not have none. They have some.
Right.
So he said, I would expect that fishes could be infected with retrovirus. And it's in fact true. There are fish retroviruses, endogenous retroviruses. They can be traced just like any others. So it's not that they lack them. I'm sorry if we gave you that impression. Number 2, I'm a non-traditional student who will attend the Cleveland Clinic Lerner College of Medicine this summer, returning after almost 12 years as a research medical device engineer.
Cool.
He's going to medical school. Wow. It's a 5-year master's MD program that focuses on research as well as medical training, and I'm very excited. CCLCM's mission is to create physician investigators, and as such, I'm keen to learn more about potential virology or parasitology-related research career paths with a formal medical training beyond the obvious infectious disease specialty. Any thought would be appreciated.
Good luck.
Well, you could go into research. You can do a research residency afterwards. Instead of being an ID'd physician and treating patients, you can do research on infectious diseases. So if you don't want to just do clinical career, I would do that. And Scott Hammer here knows an awful lot about that. If you want, I can pass you on his email. He's very good at advising physicians how to do that. Number 3, just wanted to comment on Alan's mention of microfluidic devices many podcasts ago. He brought up the possibility of lab-on-a-chip products for doing complex but predictively repetitive lab techniques like PCR. As a former microelectromechanical systems engineer working with microfluidics, I had to smile. While the field looks promising, I think it is safe to say it has yet to live up to its hype. Given the amount of time and money invested in the core technologies of MEMS, it is my opinion that beyond very specific applications, such as accelerometers in your kid's Wii and your car, Just fallen flat on its face. If I had to guess, I would bet that a novel and disruptive technology will obsolete the lab on a chip within the next decade or so. Now, that, of course, is just my 2 cents, but I couldn't resist.
I would mostly agree that this is a field that has been just about to produce the next breakthrough for 10 years or so. Um, but it is very interesting to me, having come out of the lab, to see these super miniaturized prototypes. And the possibility is there. It's definitely, though, good to point out this is a research grade technology. This is not something that's going to be in every cell phone tomorrow.
But microfluidics has revolutionized sequencing, right?
It certainly has. That's a good point. This is something, for researchers, these are devices that are already in use. So lab on a chip devices are available. They're available off the shelf. They're routinely used. But I think what I might have alluded to was the possibility of, people have been talking about using these as diagnostic tools in the clinic, and manufacturing them cheaply enough that they could be distributed much more widely. That is what still has yet to
Right.
happen.
Got it. Yeah, the microfluidics has made it possible to rapidly change the reagents and do very
Right.
quick sequencing. It's very neat. And finally, if you haven't seen the following basic science research publication, I just think it's really cool. It's a paper that details the detection of glycine of extraterrestrial origin that was captured from a sample taken in 2002 from the comet Wild 2. What is personally very exciting for me is that I've known the lead investigator, Dr. Jamie Elsila, since high school. We're both members of a radio astronomy team, RAT, that built our own radio telescope. That core group of geeky high school students have all gone on to successful and interesting engineering and science careers. RATS is a poignant example of how early mentoring and exposure to advanced sciences can have a direct impact on society in very positive ways. Did you know about this glycine discovery, Alan?
No, I hadn't seen this, but it's very, very cool.
Yeah, they got it from a Stardust probe
Yeah.
from this comet, and it's got glycine on it.
Yep.
So it says glycine has been detected in meteorites before, but the Stardust find is described as a first in cometary material. Interesting. Well, that's pretty neat.
Yeah.
Thanks for sending that. And you're right. I think if you get pretty advanced science at a young age, it could make a difference in the right students. Not everyone, because a lot of them don't respond at all, but obviously in your case. And clearly, since your name is Vincent, you were primed for that. But thanks. Sorry, he says, sorry for the long email. Keep up the good work. I look forward to every episode. No problem about the long email, but it will be our last. We will now move to our picks of the week, and there will only be two.
Because there are only two of us this time.
Right. Go ahead, Alan.
Well, I toyed with a couple of other ideas, and then I realized there's something, a site that just went up a few weeks ago that is somewhat relevant to one of the discussions we had today. The site is data.gov, data.gov, and this is a site. There's been a lot of talk about government transparency, and finally, here's an initiative that really takes some meaningful steps on it. So this was launched by the Obama administration, I guess about a month ago, and this is a central clearinghouse for databases and data sets of all different kinds across the entire U.S. government. And part of the launch of it was that each major agency was required to put at least one new database that hadn't previously been accessible online. So there's a whole bunch of stuff here, and it's elegantly designed, fairly well organized, and you can search the whole darn thing. So you could just type in your term, go looking for something. I'm just trying a couple of terms here. Oh yeah, I just did virus.
Yeah, I did that too. Infectious hematopoietic necrosis virus.
Right, or if you want the data from the 2009 flu season, and look through that data set. But that's just one example. If you're interested in census data, if you're interested in business data, or information that was collected by local and tribal governments. There's just a massive, massive amount of stuff here. And they are continuously adding to it. So the plan is that they're eventually, I mean, if it lives up to its promise, we'll see. But I think this is, it's certainly a step in the right direction. There's a whole section of geodata if you need mapping information, and a lot of it is very, very technical stuff. I don't even know what the what the hydrologic units of the United States would be, but you can download them. So all that
Cool.
stuff is online now at data.gov.
I guess, yeah, there's not a lot of virus data yet, but I suppose with time there could be more, but maybe that's not going to be
Right,
the
and this
focus
is
of this.
really, it's focused on data sets and databases, so it wouldn't be a publication
Right.
collection. And I think they were not trying to duplicate things like genome databases. So for a lot of the sciences, we already have access to databases online. This would be for things that, maybe you didn't think of, or you wondered if you would ever be able to access. It could be very useful for public health. There's a lot of population level data in there.
Yeah.
Um,
Yeah, it's very cool. So this is data that people want to make public, or it's governments
it's
supported,
government.
right?
It's things. Information the government has collected.
Mhm.
Um, and previously, it was either you had to drill down through the quirky little individual departmental websites to exactly the right place, or you had to know the right people. In some cases, it wasn't even available at all, I gather.
I see. So there's no
So,
CIA data on confidential stuff here, right?
I think if you go to that part of the site, they have to kill you.
Okay. With that warning,
Yeah.
all right. My pick is suggested from Howard, who wrote, I'm a biochemistry chemical engineering major at the University of Ottawa, and I just started listening to your podcast, and I'm almost caught up. I had a recommendation for pick of the week, a free game called Fold It. It's a fun puzzle game and a great way to learn about proteins and how they work. So I'm picking this because we have recently picked a couple of distributed computing websites, SetEye and Fold at Home and so forth. And we may have mentioned Fold It before, but it's pretty cool. It's they're basically games in which you can download, and they're individual puzzles essentially, which in the case of solving them, you're doing protein folding problems. And so they're using your computing power to figure out information about how proteins fold.
Well, and they're using specifically your own brain's computing power.
That's right. That's right. It's a
Because
game.
these are problems that are computationally very hard, but that humans have a somewhat easier time solving. And so you can play the game and contribute to science by actually putting your brain to work.
Right. So they give
It's
you
very, very
a
cool.
problem, and they say, okay, pack the proteins in the best way you can, the smaller the protein, the better. More precisely, avoid empty spaces in the structure of the protein where more water molecules can get inside. And so if you go to Fold It, actually it's fold.it/portal, they'll tell you how to do it. You download a little app. It's either for Windows, OS 10, or Linux. Actually, very cool. So thanks, Howard, for
Neat.
sending. I wanted to tell you, Alan, I was in a plane. I was coming back from Scotland a couple of weeks ago. And, there's a monitor in front of your seat where they show things, right?
Right.
Movies. And at one point it crashed. And they rebooted it, and it was Linux. It was running Linux.
That is very, very common in embedded systems these days.
Really? I saw
Yeah.
the boot up sequence. It was just lines and lines of text, and you could see that it was Linux. I was
Yeah.
just blown away that an airline would do that.
Well, no, that's for systems that are built in where they want as little maintenance as possible, and they want as much reliability as possible. A lot of these things are now running Linux. If you have a Wi-Fi router,
Mhm.
it's probably running Linux.
I know that in the airport itself, the departure and arrival screens usually run Windows because they crash periodically, and
Right.
you can see the blue screen of death on
Right.
every monitor in the airport. It's so funny.
Right, because that's running on a PC somewhere, so they use the default PC operating system. But for things that are actually built into a setup where they're not plugging a PC in, but they're just putting a black box in there and wiring it up.
Yeah.
The Linux has pretty much taken over, just as it has with the server market.
Yeah, it was very cool. I meant to
Yeah,
mention it, and I thought our listeners might be amused.
cool stuff.
And that will do it for TWiV. You can find Alan at dovedocs.com, and I think also alandove.com.
I now own the domain alandove.com. I'm not sure why, but I do.
I just tried it earlier, and it goes to DoveDocs.
Yes.
So that's good. You now own your own domain. It's very good. I've had Racaniello.com for some time, but I haven't done anything with it. And if you type Racaniello.com, it says home user page unknown. I should have it linked to something.
Yeah, put a redirect to TWiV or something.
I was thinking of making a aggregating page where there's everything that I do online on it.
That's an
my
idea.
podcasts and other stuff that I have, but it's it takes some time to do that.
Then you have to maintain that.
Yeah. So anyway, Alan Dove, dovedocs.com or alandove.com. Thanks for joining me today.
Always a good time.
And as always, send your comments and questions to TWiV at twiv.tv. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.